Good afternoon, class. Today I will be talking to you about a revolutionary gene editing technique, and that is CRISPR-Cas9. Imagine having the power to edit our genetic code, potentially curing genetic diseases. Today I'll be explaining the fascinating components of CRISPR-Cas9 and how they work together to revolutionize gene editing. As someone with a background in biology and a keen interest in genetic research, I'm excited to share this groundbreaking technology with you. So in the next few minutes, I will break down the essential components of CRISPR-Cas9 and their roles in gene editing. CRISPR stands for clustered regularly interspaced short palindromic repeats, a gene editing technology derived from the natural defense mechanism of bacteria against viruses. It utilizes unique spacer sequences from past viral infections and incorporates them into the CRISPR region alongside repeated DNA sequences. The CRISPR region in bacteria contains repeated DNA sequences interspersed with unique spacer sequences. So these spacer sequences act as a molecular memory, allowing the bacterium to recognize and respond more efficiently to recurring viral threats. CRISPR's significance lies in its ability to serve as a memory of past viral infections, so acting as an adaptive immune system for bacteria. This natural process has been harnessed by scientists to create the revolutionary gene editing tool CRISPR-Cas9. By modifying the guide RNA in Cas9 nuclease, researchers can now target and edit specific genes in various organisms, with significant potential applications in medicine and agriculture. Guide RNA is an essential component of the CRISPR-Cas9 gene editing system. It is an engineered RNA molecule that guides Cas9 endonuclease to the specific DNA sequence of interest for targeted gene editing. The structure of gRNA includes two components, which is the CRISPR RNA and trans-activating RNA, which is the target RNA. The cRNA carries a customizable sequence that complements the target DNA, while the tracer RNA guides Cas9 and forms a stable complex with the cRNA. In some cases, these components are combined into a single chimeric gRNA. The gRNA's customizable sequence directs Cas9 to the precise DNA location of interest. When the gRNA Cas9 complex binds to the target DNA and recognizes the protospacer adjacent motif, hence PAM sequence, Cas9 cuts the DNA at that specific location. So this cut initiates the natural repair mechanisms leading to specific gene modifications or insertions, enabling precise gene editing in various organisms. Cas9 is an endonuclease, which is a type of enzyme that can cut DNA. It plays a crucial role in the CRISPR-Cas9 gene editing system. In the CRISPR-Cas9 system, the Cas9 serves as a molecular scissors. It is responsible for locating the specific target site within the DNA and then cutting the DNA at that precise location. So PAM is a short DNA sequence and it is essential for guiding Cas9 to the correct location. So it highlights the specific sequence that is adjacent to the target site so it can recognize and bind to the DNA before making the precise cut. So the presence of the correct PAM sequence ensures that Cas9 only acts at the desired location, enhancing the specificity of the gene editing process. So applications. When it comes to gene therapy, for instance, um, correct genetic defects, and it can also, for example, treat diseases like sickle cell anemia or cystic fibrosis. Um, so crop improvement when it comes to agriculture or growing food, or making sure it has the right nutrients or how fast it can grow. And, for example, the US is a huge nation and have plenty of people to feed. So CRISPR-Cas9 can be a perfect tool to utilize when it comes to crops, as well as disease modeling. So it can help us introduce different mutations, correct mutations to correct, the non-working gene, for example. So we can use it to test drugs. We can use it to test on animals in many ways where we don't harm anyone or anything. Um, CRISPR-Cas9 is actually a diverse tool. It can do many things. And for instance, medicine, agriculture, and biological research are just the three main points I decided to bring up. So, there's ethical considerations and regulation, of course, are crucial as we explore the possibilities and implications of this powerful technology. So, for example, people will talk about designer babies, and this is implementing that. Um, you can design your baby to have like blue eyes, dimples, whatever the case may be, but this also comes with having money. So if you want to do something like this is also very possible for millionaires, but this is one thing that people bring up when it comes to not funding research regarding Cas9, which is like a 1% thing that someone would want to do that. But we were trying to use Cas9 for genetic diseases. The main other thing is its access and affordability. It is very expensive and not many people can access it. So CRISPR-Cas9 is a powerful gene editing tool that utilizes key components, CRISPR arrays, Cas9 enzyme, and guide RNA to target and modify specific DNA sequences. So with a better understanding of these components, we can appreciate the enormous potential of CRISPR-Cas9 in advancing medicine and our understanding of genetics. Thank you for joining me, and have a great night.